Scats are most often stored dried in preservation buffer or frozen. DNA from scats can also be obtained employing the swabbing technique. DNA recovery rates vary among species. Therefore, a preliminary trial to determine the best storage and DNA extraction methodologies is highly recommended before using scats as a DNA source. Pick up scats with clean forceps and place them in a vial containing a preservation solution such as ethanol, DMSO, and in some instances lysis buffer. Once in preservation solution, samples can be kept at room temperature away from light. Ethanol is widely used when air travel is not involved. If samples do have to be transported by air, lysis buffer may be a better option over DMSO, as it doesn't have the increased health and safety risks of DMSO. Scats can be kept dried in Ziploc bags with silica beads. Simply place the Ziploc bag inside out and use it as a glove to collect the scat. Then add silica beads to the bag. Silica will absorb the moisture, thus avoiding DNA degradation. For both the above techniques, the recommended amount of preservation liquid or silica is between 5 to 10 times the sample volume. Scats can also be sampled by swabbing. Hydrate the swab with either sterile water or a preservation solution of your choice. It's very important that the preservation buffer or sterile water come in contact with only clean new swabs. If a scat is fresh, hydration of the swab may not be necessary. Proceed to swab the outside of the scat. Allow the swab to dry and place it in its protective case and keep it at room temperature, or if available, inside a fridge. When collecting scats, it's important to know or at least infer information of the biology of the animal. For example, goats or rabbits excrete pellets, so that if a clump of pellets is found, it could be assumed that they came from the same individual, and therefore be stored together. If in doubt, always collect scats separately. It's important to remember that a pilot study is highly recommended before any genetic-based project is carried out. Such pilot study should compare different storage and extraction methods for material from your species of interest to ensure that enough DNA will be recovered to amplify successfully.